Now, the main lab activity for the endocrine system is the lab that we are doing on the human menstrual cycle. And the reason why we are actually doing this lab is because, first of all, it really demonstrates the process of negative feedback among several different hormones very well. In addition, because it involves so many hormones, it gives us a chance to look at how several of them do function and a little closer look at what they do. Now, it is very, very important that you follow the procedure as it is written and that you answer the questions as you go along. What tends to happen is people jump ahead to the different parts of, like the graphing, for instance, that I'm going to explain here in a minute, and they get all of the graphing done, and then they go back and try to answer the questions. And for some of them that works, but for some of them, if you don't have only what is supposed to be graphed at the time, it can be very difficult to try to answer those questions. Now, if you look at the back of the front page, you actually see four different diagrams. So you have this one in letter A, okay, this graph in letter C, and you can see then that they have underneath letter A, they have B, which shows some picture of what is actually going on in the ovary, and then underneath letter C, they have a correlation to what's happening in the uterus, and that is actually how we want to look at these. So let's go back here to letter A, because that's first what you're going to be graphing with. And if you look at number 2 in the procedure, it says using the data in table 1 on page 176, plot points in part A of figure 1. Okay. It says, remember to use the left-hand vertical axis. So that's the first thing I want to show you, is that you actually have two vertical axes. We are going to be using the one here on the left, which monitors FSH. And if you look at table 1, it takes FSH on days 1, okay, through 14. So I'm only going to be plotting on this for this portion of the line here. So I need to use this as my y-axis and this is my x-axis for that segment. And then I have two questions. Now, the other thing that it does say in the lab is it says use a pencil to draw a curve. And it says do not just connect the dots, draw a smooth curve. Go ahead and just connect the dots. Okay. After you've connected the dots, answer questions A and B. Okay. Please make sure that when you answer letter B, okay, it says what happens to the follicle. So you need to compare what your graph is saying with what you see is happening here and what is written in the kind of background information above the number 2 on your lab. Then you go to number 3, which says in part C. So now I'm going to be down here in part C. Use the table in data in table 1 to plot the points indicating concentration of estrogen. So again, I'm going to be on using the left-hand side axis here for days 1 through 14. Okay, it says draw a curve. Remember, we are connecting the dots. All right. Then it says to look for number 4. It says look at part D. Okay, and then it has a question there. What happens in the ovary, part B, and in the bloodstream, part C, that brings about the change in the uterus? So you have to look at part C, what is happening, and also in part B, and try to figure out why we see the changes that we see here. Okay? And so this is what I mean. You have to take this very, very slow. Eventually, you are going to actually have two different lines for both graphs, and both lines are going to span the entire distance from this left-hand side all the way over to the right-hand side. So you will have one line here that represents FSH and another line here that represents LH. So when you reach the part of the directions where you start plotting LH, please make sure that you are using the right hand scale to plot that. Okay, and then when you get down to progesterone, that you use the right hand scale to plot that. The scales are different, and if you try to plot the progesterone information using the estrogen scale, it's not going to work. Okay, because you have two different lines, you do need to label them or somehow make them different. My suggestion would be to plot your lines first using regular pencil, and that way then if you make a mistake, you can go back and erase it. If you just want to draw a line or an arrow or something pointing to the line to label it, that is fine. If you want to add color, so make one line one color and one line another color, that is fine. What I would do is I would go back after I'm sure my line is correct and do that color either in marker or colored pencil. Colored pencil doesn't erase very well, so please make sure that everything is correct before you do that. Okay, then at the end you have some analysis and interpretation questions that are going to take you back to looking at the graphs and the information, and remember, those should be answered on your own. In addition, some of this includes information that may feed you back to the background information and your background knowledge of feedback mechanisms that we have already discussed. So you may have to go back to Unit 1 and look at the information about negative feedback and positive feedback and see how they fit into the picture.